But we're doing chapter 15, which is pathogenicity today. So what does that mean? If you've done your oral presentation, what is pathogenicity? How would you define it? Yes. So inside the cell, like how is it getting into your body as a part of it? That's transmission. But once it's sort of in the cell, in the host, whatever, the initial host, what happens next? And usually it's focused on really a very tiny level, right? So we're thinking, we're not like, oh, does it cause a fever? Does it cause a rash? Or did you get it from eating contaminated beef? Or it's a bacteria. Pathogenesis and pathogenicity has to do with what is happening. Like, what are the physical components of the causative agent that are causing, how is it able to get into your body? Why does it give you those symptoms? What is happening to your cells, and what is it about this causative agent that is doing that stuff? Does that make sense? So yes, pathogenicity, really, strictly speaking in the narrowest sense, you're right, could be just a reference to how bad is it, how virulent is it. But really we're sort of talking about the broader picture of the process of the disease and what are the pieces of the microbe that are kind of getting you sick and exactly how, right? Because every microbe's different. Clearly, the influenza virus is getting into your body and causing a different kind of cellular problem than, say, gonorrhea. You would clearly say that those things are different. But why are they different? What are the actual components of the pathogen that are making that different thing happen? So we are going to talk about, when we say mechanisms of pathogenicity, we're sort of saying, what is, if we go zoom in on that microbe and we really look at it in detail, what is it doing? Why does gonorrhea cause this particular kind of STD and the flu virus gives you a respiratory infection? What's happening? So pathogenicity itself, that word means the ability to cause disease. So this question, hopefully you all have a good answer to, but are all microbes pathogens? No, right? Many of them are harmless or some of them are helpful. Virulence is the extent of the ability to cause disease. And we're going to talk about two important terms, okay? And you will have to understand these, be able to use them. They may well appear on a test question, and so you need to understand them well enough to problem solve, basically. Yes?
So when you say virulent, is it caused by viruses?
No, and that's a great question, and it's confusing, right? So virulence has that viru part of the word, but it is not referring just to viruses. Virulence means, think of it as sort of strength, if that's helpful, the strength of the ability to cause infection or disease. So you could, for instance, here's an example, there might be two strains of flu, and one makes you much sicker. It's more virulent. Another example would be if HIV evolves and it's much more effective at being transmitted or something like that, then it's more virulent. If an organism acquires some new molecular trick and it makes you sicker or it's more effective at getting into your body, that would increase its virulence. Does that make sense?
Yeah, but the examples that you said they're all caused by viruses, like HIV and other.
Sure. Okay, I'll give you another example then. So do you remember the classic experiment a lot of people talk about when they talk about Koch's postulates, those rules of how to establish this is the pathogen that causes this disease? And one of the things that people often talk about is that experiment with the mice, where you inject the mice with the regular virulent strain of bacterial pneumonia and the non-virulent strain. Do you remember what the difference was between the harmless one and the harmful one? It was the capsule. So the capsule made that strain virulent. Does that make sense? Okay. So it is not just viruses, it's bacteria. It could be, anything really that causes disease. So these two terms are ID50 and LD50. And ID refers to infectious dose, and LD refers to lethal dose. And they actually refer to two different scenarios, kind of, while they're related and they could be used to describe the same microbe, they refer to different things. So the ID50, or the infectious dose, is basically the dose that causes infection in 50% of a population. So let me break that down. First of all, dose really means number of microbes, okay? So if you're talking about a virus, then dose would be what? Number of viral particles, okay? If we're talking about a bacterium, what does dose refer to? Number of bacterial cells. It's very simple, okay? So this is the number of the microbes of that particular species that would, if introduced into some experimental population, make half of the hosts sick, okay? Now, this is going to be experimentally defined or contextually defined. So, for example, let's suppose that you take a population of super healthy, fit, 21-year-olds with no stress and, you know, lots of free time and plenty of nutrients, and you expose them to, I don't know, some flu. Now let's take a similar number of elderly people and babies with no immune system, and maybe some patients who are immune compromised, and expose them to the exact same number of microbes. Which population is going to have a higher level of illness, most likely? Most likely this other population. So if you're looking for the number that's going to make half the population sick, the exact same microbe, that number is going to be different depending on the sample that you're exposing it to. So it's going to vary. Like if you take the exact same strain of flu or the exact same bacteria, then the ID50 is going to vary depending on the population. So it's always experimentally defined. It's always defined by the context. LD50 is the lethal dose, and it refers not to number of microbes, it refers to amount of toxin. So it's always talking about a toxin. Now, are all microbes producing toxins? Are all pathogens producing toxins? Is the way that you get sick by some infection always a toxin-based effect? No. There are definitely some very classic examples of things that make us sick with toxins. Does anyone know of any examples? Say again?
Toxic shock syndrome.
Yeah? Toxic shock syndrome, or E. coli 0157H7. Remember, it makes that Shigella toxin. Or botulism, botulinum toxin, or what's another one related to botulism? Plus same in Clostridium genus. Tetanus, yeah. Okay, so there's lots of them. There's also gram-negatives that have that endotoxin, so you could refer to that. Usually we're talking about an exotoxin, but it's the dose of toxin, so the amount of the substance itself. If you were able to sort of chemically extract it and measure it, it's usually measured in some amount per mil, a concentration. And so again, it's the amount of toxin that if you gave it to this population, half of the hosts would die. And do you think that, like ID50, LD50 is also defined by context? Yes, okay, so maybe not in every case. There could be a situation where, you know, if it's a human pathogen, then no matter who you were, you would be equally likely to die, but it's very unlikely. It's most likely the case that many factors, like whether you smoke and whether you're 12 or 27 or 100, those things will all affect the lethal dose. Okay, so for any given microbe, the microbe's the same in different experimental situations. Is the ID50 going to be the same in every situation? No. You have to actually say, this is what we found, and now we can say, so you take the data, and now you say, okay, now I know what the ID50 is in this situation. Now, let me ask you this. Which is less likely to cause disease? A microbe that has, in some particular situation, a higher ID50 or a lower ID50? Yes. Lower ID50 means that in this particular situation, it takes fewer microbes to make half the population sick. Does that make sense to everybody? So a smaller number, a smaller ID50 means that it's more infectious. Now, the same kind of thing applies to toxins, and here's just some examples of botulism toxin, botulinum toxin, Shiga toxin, and Staphylococcus enterotoxin. All of these are exotoxins. And these are the lethal doses that are just on average over many different exposures and many different populations. On average, we can come up with a lethal dose. So if this was just the numbers that you were given, 0.03 nanograms per kilogram. So what does that mean? So you can measure it in amount per weight or amount per volume or however you do it, but usually you're doing it like the kilograms would refer to the weight of the host, and the nanograms would refer to the amount of the toxin, okay? So if you're just measuring how much toxin it takes to kill half of the population and you're doing it by nanograms per kilogram, then a very small amount per kilogram of botulism toxin will kill half the population. A larger amount of Shiga toxin will kill half the population, and almost 10 times that for Staphylococcus enterotoxin. So of these, which is the most potent toxin? Botulism, right? Which we already know is one of the most potent toxins. Okay, so there are many things to consider when we talk about pathogenicity. One of the things that we think about is how does the organism get into your body? And there are several portals of entry, and every organism's route is going to be specific, and there are going to be preferred ways. So for instance, just as an example, if you are infected with the flu virus and you're standing next to someone and you sneeze into their mouth or sneeze onto their hand, which is more likely to make them sick? Into the mouth, because we know that inhalation and mucous membrane entry is how that virus is transmitted. If you get it onto your skin, as long as you wash your hands, you might be protected. It's certainly not going to go through the barrier of your skin, unless you wipe your eye or lick your hand, as one is prone to do all day long. Now, there are some organisms that have only one route of entry and some have multiple. So here is a couple of examples of ones that have multiple routes of entry. So Yersinia pestis, which causes the plague, has several different routes of entry. Bacillus anthracis, which causes anthrax. You can get it on your skin, you can get it by inhalation, you can get it by ingestion. There's many different ways. And depending on how it gets into your body, it can cause different symptoms and a different level of severe illness. There are plenty of examples, as I talked about with the flu example, where an organism that makes you even very sick by one route of entry is harmless by another. So, for example, Salmonella typhi causes salmonella. How does that organism get into your body to make you sick? By ingestion, okay? If you get it on your skin, okay, if you're preparing food and it's on your skin but you never put your hands in your mouth or you wash your hands, then are you going to be sick? No. The bacteria that causes pneumonia, because pneumonia has a number of different possible pathogens, like there's viral pneumonia and fungal pneumonia and bacterial pneumonia. The bacteria that causes pneumonia causes it if it gets in through your respiratory system. But if you eat it, right? I don't know what kind of weird example, but let's suppose someone sneezed on some meat. I know there's a lot of sneezing happening in my examples, and I'm sorry. And then you held your nose and then you ate it, right? When you ingest that particular pathogen, your stomach will kill it, so you won't get sick. So how do microbes get into your body? There's a number of ways. Obviously, we know the mucous membranes is one route of entry. So that would be the gastrointestinal tract and the respiratory tract. These are the two most common ones. So we can eat things that cause us illness by getting into our stomach lining or into our intestines. We can inhale things like the flu or the cold virus or some bacteria. Less common, but also important, the genitourinary tract, as well as the mucous membranes of your eye. Some organisms do cause an illness if they get on your skin. The skin is a major barrier to to entry. It's mostly inaccessible to pathogens, but there are some openings in the skin, like hair follicles and sweat glands. And so, for instance, some parasites like the hookworms can actually bore right through your skin. So you just get it on your skin, especially it tends to be on your feet. And and they're tiny, so they wiggle all the way up through the layers of your skin and get into your body that way. We also probably know that you can get like toe funguses and funguses on other parts of your body. So there are fungi that can grow. And usually the fungi will just grow on the surface layer, or they'll get through the layers slowly, and they'll just be eating the protein in your skin cells, the dead cells. They're using that as food. There's also this route, the parenteral route, which is not the same as parental. Okay, this is not a PG-rated movie. What does parenteral mean? That is when your skin, which is usually a really good barrier, is cut. Okay, so under what circumstances would this happen? How could you get an infection via the parenteral route? What are some things that would qualify? So we have to pierce or cut the skin. How does that happen? Say again? Yeah, so puncture wounds. Yes, absolutely. Any other things, like what if you're in the hospital? How could you get a parenteral infection? Say again? Yeah, so you have IVs. Also, you've got surgery, right? So if the surgical instruments aren't properly sterilized, or the air has contaminants, or, as your wound is healing, things get in there. So it's the penetration of the skin. This also includes penetration of the mucous membrane. So for instance, catheters would be an example. And it also includes insect bites. So cuts, even like a paper cut would qualify if that caused an infection. Okay, so just some examples of how to use ID50 in particular. This is the bacteria that causes anthrax. And I mentioned before that it has several routes of entry, and depending on how it gets into your body, it causes a different severity and a different kind of illness. But what you'll also notice is the infectious dose varies depending on how you're exposed. So, if Bacillus anthracis spores, is how it's really passed, if they land on your skin, it only takes between 10 and 50 of these endospores to make you sick through your skin. Or another way to say that is to infect half of the population of any given average population, it takes between 10 and 50. Okay, why is there that range? That range is there because maybe this is a population of 75-year-old people, or maybe this is a population of patients in a hospital, or maybe these are teenage, kids with no other health problems. But between 10 and 50 endospores on your skin will give you anthrax. On the other hand, if you inhale it, let's suppose you receive, I think that enough time has passed for me to make this joke. If you receive some powder in the mail and you decide to snort it instead of calling the authorities, then you have to inhale between 10 to 20,000 endospores to get sick via this portal of entry. Or if you decide that it looks like some sort of spice and you put it on your food, it takes even more. So of these different methods, which way is Bacillus anthracis going to be most virulent? Yeah, if it's coming into contact directly with your skin. So your best protection, if you're handling weird powders, is by wearing gloves, okay? And then calling the authorities and not snorting it or eating it. But this is going to be different depending on, obviously if we're talking about flu virus, completely different, right? Of these, without knowing what the numbers are, but of these different routes of infection, which one is the one that's going to be most effective for flu virus? Going to be inhalation. Ingestion will do nothing. Getting it on the skin will do nothing, right? Unless you wipe your eyes or whatever. Okay, so bearing in mind that the three main portals of entry are what? They're the skin, mucous membranes, that's basically GI tract, genitourinary tract, and conjunctiva. As well as, what's the third one? The PG-rated parenteral. So of these, which one is malaria? How do you get malaria? Insect bites. Okay, so of these, what is your portal of entry? It's this one, right? So insect bites or puncture wounds, that would be. How about something like tetanus? How do you transmit tetanus? Also parenteral, generally speaking. Okay, what about botulism? It's going to be this one, right? What about Bacillus anthracis? Most likely, skin. Okay. Influenza virus, mucous membranes. Same with HIV, right? Mucous membranes. Or, potentially could be parenteral if it gets into your bloodstream. And then Bacillus anthracis actually has, all three of these are possible, but most likely it's going to be the skin. So why is it that these different organisms have different routes of entry? Why do you think that is? Say again? Yes, that's exactly right. So they have, whether it's a virus, a bacteria, whatever it is, they have things on the surface that are going to contact and bind specifically to different cell types. So if you're a flu virus, your ability to bind to or adhere to something, you're only going to be able to bind to respiratory epithelial cells. If you're, say, E. coli, right, you're going to be able to bind to intestinal cells. So it's just a matter of you have only certain abilities to bind to cells. And if you, say, are E. coli and you get on skin, there's nothing there for you. Okay, there's nothing that you can do. You don't have the right keys to get into your targets. So it does come down to some extent to adherence, the ability to actually, here's the pathogen, here's its adhesins, right, things that it's going to use to grab onto its host. And what it's grabbing onto is some receptor on the surface of the host cell. So this is the plasma membrane, and the rest of the cell is all out here. And there's some receptor that it's binding to. And if it's some other cell type, this receptor is missing, so it can't do anything with that. So these adhesins or ligands, as we call them, on the surface of the pathogen bind to host cell receptors. Where are these adhesins located? They might be in the glycocalyx, the fimbriae, the pili, or the flagella. All of these are things that are sticking out of pathogens, right? Or they could be spikes on the surface of a virus. They're usually glycoproteins or lipoproteins. So what does that mean? What is a glycoprotein? Yeah, it's a protein decorated with sugars, right? And remember that when you put proteins into your membrane, you decorate the outside with those sugars, and that becomes part of the glycocalyx. Receptors typically have mannose in them. And this is important, right? And this sort of just brings home my point from earlier. Both these adhesins or ligands on the pathogen and the receptor that they're binding to, this is the specific interaction, right? But it could be anything. You could just change the shape of this and the shape of that. As long as they bind to each other, that's all that matters. So you could have altogether different adhesins and different receptors, and then that means it's going to be it's going to target a different set of cells. So what is the basis of pathogen specificity? Why do pathogens target only certain species of hosts and only certain cell types within a multicellular species? Because they have only the ability to bind to certain specific things. Now, there's also this factor, which is the ability to adhere. And so in this context, biofilm formation is important. So it's helpful to recognize that, when we're doing, like, say, labs, we're dealing with pure cultures. And as I've said before, in nature, microbes typically live in a community. And in your body, which is sort of a natural host for pathogens, about 65% at least of your infections are actually biofilms. Okay, so the species that's causing an infect is in a community, and all those cells together are working together to cause an effect, and maybe working with other species as well. So for instance, if you look at dental plaque, there's several species, and they're creating together that community of cells is greater than the whole, or than as individuals. Because together they create this biofilm, this kind of slimy coating that protects them. When implants that you put into the body become colonized, then they can cause infection because of this biofilm formation. This is also why, for instance, those of you who wear contact lenses, sterilize them on a regular basis, and if you don't, horrible things can happen. If you read the news on occasion for the horror factor, very bad things can happen. And that's because over time, your contact lenses accumulate biofilms, which are just microbes growing on there and secreting stuff that's not good for your eyes. So there are a number of ways that pathogens get past our natural defenses. One of the ways that we've already talked about at length is, does anyone know what is one of the chief ways that pathogens can avoid being engulfed by white blood cells? What gives them kind of a cloaking device, makes them invisible? Capsules, exactly. And so other cell wall components, and besides the capsule, are important in establishing an infection. The other thing that's really important is antigenic variation. Now, what the heck does that mean? What is antigenic variation? We've mentioned it before. Does anyone remember kind of any of the components of this term? Does anyone remember what an antigen is, which we will come back to in the immune system section? What about variation? Yeah, there's like different antigens, yes, and we create antibodies in our immune system against antigens, and then if they change, then our immune system can't recognize that thing anymore. So some microbes, some pathogens are good at changing their antigens, and then they are faster evolving new strains that are not, that we have no natural immunity to. There is also an involvement sometimes of the host cytoskeleton. So you'll remember that eukaryotic cells like ours have some filaments inside of them that organize stuff. And some microbes will actually hijack that cytoskeleton for their own purposes. We'll talk about that. There are also extracellular enzymes that become important. So let's talk about capsules first. They basically prevent white blood cells from engulfing a microbe. So they allow them to escape. And here are some examples of species that do this. So these are bacteria. Streptococcus pneumonia, so that's the pneumonia bacteria. Haemophilus influenza causes a bacterial flu. And Bacillus anthracis. All of these are encapsulated. If you took the capsule away, you would reduce or eliminate its virulence so that you would have a strain that's in every other way identical but does not make you sick. And that's just because our immune system suddenly would be like, oh, well, there's a bad guy, let's get rid of him. So, for example, if you take the capsule away, bacterial pneumonia is completely harmless. Now, it's important to just say this. There are lots of things that are not human pathogens that do have capsules, okay? So having a capsule will be helpful, but doesn't mean that you're a pathogen. All right, we're going to talk about two specific cell wall components. One of them is the M protein. This also helps an organism avoid being engulfed by white blood cells. We talked about this, or we will talk about this again in chapter 16. So this is just a note to remember this concept for later. It's a protein that's found on fimbriae, these projections that come out of bacteria. And some examples of diseases that are relevant to this M protein. So Streptococcus pyogenes, does anyone remember handling this back when we did that, some of those plate tests in like lab 3? Streptococcus pyogenes, do you remember anything about it? Like what plate we put it on, what the effect of that was? No? Do you remember your blood agar plates? do you remember that there were 2 strains that we put on those plates, and one of them did what? Beta hemolysis, and the other one didn't, right? So this is that organism, right? This Streptococcus pyogenes that when you put it on a blood agar plate explodes the red blood cells. In your body, it will do the same thing. There's tons of strains of this that cause different illnesses. So some strains cause strep throat, others scarlet fever, toxic shock syndrome can be caused by this strain. And it's got M proteins on the surface, so it makes it hard for your immune system to engulf those cells. So here's just a picture of that, an electron micrograph. Here's the cells, and if you look at the fimbriae that are coming out of it, this is done with an antibody, that's why these little dots are there. This is just detecting the M proteins. And if you have a similar bacteria that just is not able to make this M protein, then it just has, everything else is the same except it doesn't have the M protein, and that bacteria would be less toxic. There's also something called the OPA protein, and it's also found on fimbriae that inhibits T helper cells from detecting the pathogen, and an example is the gonorrhea bacteria, or the gonorrhea pathogen, I should say, Neisseria gonorrhea, has OPA protein in it. Again, it's on the fimbriae, and it inhibits T cells from detecting it. Mycolic acid sound familiar to anyone? Have we talked about mycolic acid before? Why, yes, we have. Say again? You're actually misremembering, but in Archaea there's a different genus that doesn't have so it's a different one. But mycolic acid is this waxy lipid, and it's resistant to being digested. So it doesn't prevent your white blood cells from grabbing onto it. But once it's inside the white blood cell, normally what would happen is a white blood cell that grabs onto a microbe and takes it in, then uses digestive enzymes to destroy it. If you have mycolic acid around you, then you'll be taken in, but they won't be able to do anything about it. And so, what are the pathogens we know of that contain this? Mycobacterium tuberculosis is one of them, right? Does anyone remember any of the other ones? Do you remember Mycobacterium leprae, causing leprosy? There's 2 genuses as well. So Mycobacterium genus, all of the species in that genus have mycolic acid, but there's another genus. Do you remember that one? Nocardia, does that sound familiar from your lab right up? Not every species in the Nocardia genus have mycolic acid or are pathogens, but there are a couple. Okay, so antigenic variation, again, is this thing where a microbe might be able to avoid your adaptive immune response by changing the surface proteins, okay? And again, this is something that we'll discuss again in chapter 17 when we talk about adaptive immunity. Some good examples are the malaria pathogen Plasmodium, which is a protozoan. There's a glycoprotein on the surface, and that can be detected by your immune system, but it changes very rapidly because the way that particular sequence is treated, it undergoes a lot of changes. So there's many variants of this, even within the body. As you have an infection over time, it can change the surface, and it's as if it's changing colors or something, and you can't see it anymore. You have to re-find it. So this makes it hard for your body to fight it consistently. Some examples of bacteria and viruses with this ability. The influenza virus has a rapid antigenic variation, and the bacteria that causes gonorrhea also has this ability. So we've talked about the influenza virus, right, because it's an RNA virus. And remember I told you that RNA viruses, they use RNA-dependent RNA polymerase to replicate. And RNA-dependent RNA polymerase has no proofreading activity. So when it copies, it makes mistakes at a much higher rate than DNA polymerase and just naturally introduces mistakes. In addition, there's something special about the flu virus that causes it to change more rapidly, which is what? What about its genome is special? What about its life cycle can potentially lead to rapid changes in strains? Why are we worried about flu pandemics? Do you remember like the structure of the genome? What was special about it? It had 7 different pieces, right? And it had the ability, it has several different hosts. So if 2 different strains get into the same cell, they can shuffle all their pieces, and that can change the exact nature of the things that are displayed on the surface much more rapidly. So here's just an example of one of the organisms that has this rapidly changing ability. So this is Trypanosoma, which causes Chagas disease, also called African trypanomyosis. And it's a protozoan that has things on the surface that can be detected by the immune system, but within a single infection, during the course of its replication, there's a lot of change that's introduced into the sequence of the DNA that encodes the proteins on the surface. So over time, the most predominant sequence of protein that's displayed on the surface changes. So, initially you're infected with this guy, you develop antibodies against him, you start to knock down the levels of this, but at some point, even as this is happening, it's still replicating, and if it makes the right mistake, change, and the surface, and this guy is born, having a different protein that your immune system doesn't have antibodies against, then this starts to become more common. Then it's high enough that we make antibodies against that, and then we knock that down, but meanwhile it's been busy evolving a new kind altogether. This is all within one host. So that's why some infections can be hard for your immune system to battle. And antigenic variation also explains why there's variations in things like the flu and the seasonal flu, and we have new strains arising all the time that we then have, if we're going to do vaccinations, we have to remake the vaccine every time. And if we're talking about our body immunity, we have to sort of establish different antibody pools. So why doesn't the flu vaccine provide long-term protection? Why do we have to have a new flu vaccine every year? Because there's different strains that are no longer, we have no protection against them because their antigens are different. So when we say, okay, here's this flu virus, we know what the antigens are going to look like. Let's take not the whole flu virus, but just the antigens, put them in the people. We make antibodies against those antigens. Now this flu virus comes along, has different antigens. So that vaccine's completely useless now. All right, the last thing I want to talk about is examples of microbes using our body's cells' cytoskeleton. Usually, so the cytoskeleton, we haven't talked about this, and you may not be that familiar depending on how sophisticated your cell biology knowledge is. But inside the cell of these networks of filaments, there's 3 kinds. There's actin, there's intermediate filaments, there's microtubules. And usually it's actin that ends up being the one that's co-opted by microbes. So here's some examples. So Salmonella and E. coli are both able to produce something called invasins. They're proteins that, when they're present at the surface of the cell, they induce a rearrangement of the cytoskeleton, which is underneath the surface of the cell. And it's actually part of how the cell's shape is maintained, and it has some relationship to the fluidity of the membrane. So it basically talks to the cytoskeleton, and it convinces the membrane to do this kind of ruffling and makes the plasma membrane easier to penetrate. So the cells, these bacteria can get in more easily. There are also some really interesting bacterial species that use actin like a propulsion system to get both inside of a cell and move inside of it and also between cells. And so Shigella, Listeria, and Rickettsia are all examples of that. Here's just what a membrane ruffling event looks like. So here's a Salmonella bacterium, and it's producing these invasins that you can't see, but the invasins are talking to the proteins underneath the cell surface and convincing the cytoskeleton to change so that this membrane gets a little bit more fluid and it makes these ruffles, and that allows the bacteria to basically sneak in to the cell. This is it's an interesting phenomenon. It's a little sort of hard to conceptualize at first. So the way that actin is organized is that it's made of protein subunits that are built. So imagine there's just a little bead, that's one actin protein, and you build them into chains, and that's the whole filament. And there's lots of ways to convince actin pieces to come together like that. So some organisms like Brixedia have secrete proteins that cause these little pieces of actin to polymerize to make these long filaments behind them. So they'll secrete that protein that convinces actin to make filaments behind them, and so these filaments form and push them through the cytoplasm. This means they can get places faster than just with a flagella alone. And so, for instance, it allows them, if this bacteria is taken up by the cell, then it can make these comet tails and push itself to the other side of the cell, even pushing it out of the cell and into an adjacent neighboring cell. So it's like it's kind of like a rocket propulsion system. Okay, there's a couple enzymes that I want to mention, and I'm just going to tell you what they're called and what they do. So coagulases coagulate a protein called fibrinogen, and it basically clots blood. So Staphylococcus, pathogenic species of Staphylococcus can do this. Why would that be helpful? Imagine that you have a wound, okay, and already there's damage, and there's blood, obviously. Now, when we have things in the bloodstream coming to the site of infection, why would we do that? What is helpful about that? Say again? Yeah, and more specifically, white blood cells are coming with that blood. White blood cells are going to, find microbes and engulf them, and have antibody-making capabilities and recognition capabilities. So they're going to be coming to the site like first responders and trying to figure out what's wrong and deal with it. Now, if you have the ability to clot blood, you can prevent that to some extent, right? You can sort of make a wall. At the same time, there are also sometimes microbes that are pathogens will make kinases. Kinases digest clots and remove them. So again, Staphylococcus has this, Streptococcus also has this. Now, why is this useful? This seems like completely counterintuitive. It has the opposite effect. But what's in blood, besides white blood cells? Red blood cells, which could be food, right? Also could be a route by which you can get to another site to infect more cells. There's also something called hyaluronidase that hydrolyzes hyaluronic acid. Now, you may not know this, but a lot of the connective tissue in our body is protein that cells make outside of themselves in a big network. So they make their own network and they sit down on their own network. And if you're a bacteria and you want to get from point A to point B, you have to get through all this protein crap, and it would be nice to just digest it, make it liquid, and then you can move where you want to go. Hyaluronic acid is part of that network, and so if you just break it down using this enzyme, you're basically liquefying the connective tissue, making it much easier for you to travel throughout the body. Staphylococci do this. Clostridium has this ability, so various pathogens in that genus. Similarly, collagenase, which hydrolyzes collagen, another part of connective tissue. Clostridium particularly, pathogens in that genus do that. And this protease, IgA protease, is specifically a protease that destroys antibodies and helps you to evade immunity. So even if you have an immune response against something, but you have the ability to kill the antibodies before they get to you, you can escape. And Neisseria, gonorrhea will do this. Okay, we're going to stop here and talk about the rest of this next time.